As we've seen, we can also form phosphoesters to the hydroxyls. So this is an example of this, glucose phosphate. These are often used as activated forms of sugars in metabolism, as you'll be getting to next semester when we talk about, when Bob talks to you about glycolysis. First thing that happens is in glycolysis is you metabolize glucose, is you convert glucose to glucose 1-phosphate, and glucose to fructose phosphate, and so on. So activated forms of sugars. And of course, ATP is also a sugar ester, because we've got the base here, adenine, we've got a ribose here, and we've got a phosphoester on the carbon 5 of the ribose. So ATP is also a phosphoester. And examples occur, for example, in metabolism, both in synthesis and in breakdown of glycogen. We're probably covering this in anatomy and physiology. So we have formation, we use ADP glucose with a phosphoester to the G1 and an adenine on here as an activated form of glucose in synthesizing glycogen. Have you come across, you had a lecture on bases and the like yet, where Paul will maybe be talking about nucleotides often as activated carriers of different things. In this case, the adenine phosphates is being used as an activated carrier of glucose. That's used in adding glucose on to synthesize glycogen. Likewise, we're producing glucose phosphate as we break glycogen down. So again, we're seeing sugar phosphates as sort of center points in which carriers. Coming to our substituted versions of sugars, we can also sometimes replace one of the hydroxyls with an amine. Now in this case, we've done something quite simple. We've just put in an NH2 here, so this is fairly simple. So glucosamine with the replacement of the 2 hydroxyl with an amine, galactosamine, the same thing is happening. But this gets actually be more complex. All right, so I was going to note in passing that ATP also includes one of these, which in this case is a replacement of we have a tertiary amine on the C1 of the ribose as well, as to how we're fixing the adenine on. As I say, we can have a relatively simple amine group on, but we can put more complex things on as well. And these two, acetylglucosamine and acetyluronic acid, are amongst the monomers involved in synthesizing peptidoglycan, which we touched on before. So, this one we've got, this acetyl group is on the amine as well. This one, the uronic acid, this is the center to how the peptidoglycan works. Because you see we've got a carboxylic acid added onto the oxygen as well. And what we actually do with the peptidoglycan, or bacteria do, is they form amine bonds, peptide bonds, to add amino acids in chains onto the uronic acid. And by cross-linking those chains, it's possible to produce this incredibly robust cross-linked structure like this. So we have our sugars, our modified sugars, N-acetyluronic acid and N-acetylglucosamine this way. And on our N-acetyluronic acids, we've got peptide bonds cross-linking amino acids this way. I was meeting to some people in my marking of presentations yesterday, and I kept asking them, when they got the microscopy, what does the gram stain stain? And the answer is it stains peptidoglycan, in case you come across someone else like-minded. And the reason it doesn't stain the gram-negatives is they've got a membrane outside the peptidoglycan wall, which means that the dye can't get at the peptidoglycan. Like I say, just in case you come across someone who's as mean as me. Most of the lecture, however, is going to turn on glycosidic bonds. So, in this case, what we can do is we can carry out reactions with, to replace the hydrogen of any of the hydroxyls, and if the hydrogen being replaced is on the anomeric carbon, remember the C1 or C2 ketose, then what we have is called a glycosidic bond. This is the most common type of linkage we see in sugars. So obviously in alpha molecules you get are called glycosides. So in this case, as I was speaking about that last week, when we carry out this conversion, we take a hemiacetal or hemiketal and convert it into an acetal or ketal, which locks our ring shut. And so this is a fairly scary looking molecule, which is of some medical importance, or if you want to kill someone. This is the foxglove, very, very pretty flower. It's very poisonous. And that's because it contains this cardiac glycoside. What it does, I believe, is it makes the heart contract more, but slightly more slowly. Now, if you take too much of this, this is obviously not good, and it will cause a heart attack. But it is also used therapeutically in controlling certain heart conditions where you need those effects on blood pressure. So it is used therapeutically still, though not without some controversy. Moving on, because obviously one of the things we can do with glycosidic linkages is we can start to link sugars together. So the glycosidic linkage in carbohydrates becomes equivalent to the peptide bond in amino acids. So we can start to build up more complex structures. So we'll start with the disaccharides. So this is maltose. This is one of the breakdown products of starch. Starch is a, I will come back to later. Starch is a polymer of alpha-D-glucose. So if you're breaking it down, one of our products are different, smaller versions of different polymers of alpha-glucose, and this is one of them. Some nomenclature here. We've got obviously alpha-D-glucose. This is the full notation. There are simpler forms as well. Dash, and then in brackets, 1 arrow 4. This is showing that we have a glycosidic linkage between the carbon 1 of this glucose and the carbon 4 of this glucose. Note that I'm not specifying whether this is an alpha or a beta. I've drawn this out so that it's drawn in as an alpha here, but because the anomeric, the hydroxyl on the anomeric carbon is still a hydroxyl, this one can open and close. So it may be alpha at the moment, but it can hypothetically be formed as a beta. It's not locked, so we don't include the specification here. The arrow goes from the anomeric carbon to the other carbon that we're involved in forming this bond. And as I say, it's used quite a lot. Because if you, have you come across the mention of malting floors? In a malting floor, then, what you do is you take cereal grain, you spread it out on the floor in a fairly even layer, you give it a bit of water to get it started germinating. And so it starts to metabolize the starch, and you get malt as a result. And of course it has lots of uses. It's what's given to Maltesers, the characteristic flavour. Likewise, Horlicks, like I say, I don't get on with warm milk, so this is a nightmare for me. But I'm sure those, I think of the smell of it, that you have similar flavours to Maltesers. Uses I'm happier with. It's one of the stages in beer making. And of course, it's what gives malt whiskey its distinctive characteristics. This is my personal favourite. It's a very smoky one from Ireland. Okay, back to the serious stuff, so quiet. Lactose is another one, obviously, of importance in mammals, of defining importance in mammals. Beta-D-galactose forming a 1,4 glycosidic linkage to D-glucose. This is the main sugar in milk. So this is quite sweet, but not so sweet as sucrose. So this is why milk has this sort of slightly milder sweetness to it. And we've already spoken about sucrose a bit. Now, here you notice that our nomenclature changes a bit. our notation changes a bit. Remember I was talking about the arrow going from the anomeric carbon of one sugar to the other linkage. If you remember, as I was talking about the sucrose last week, our glycosidic linkage is between the anomeric carbons of both sugars. So in this case, we have a double-headed arrow going from the anomeric carbon on the glucose to the anomeric carbon on the fructose. Remember, the fructose has had to be drawn back to front so that we can draw this in. So we're going 1, 2, 3, 4, 5, 6 on this one. And in this case, because the fructose is not closed, because again we've got a glycosidic linkage on the anomeric carbon, we are going to specify that this is going to stay as beta glucose, as beta fructose. And I've spoken about this process in a couple of lectures already, one over in the cell science module. This is the main transport carbohydrate in plants. And the way it's done is by pumping sucrose into the phloem sieve elements, and that draws water in, and that gives us a high pressure here, a lower pressure here. So it's like pushing water through a pipe by a head of pressure. Exactly as the pump in a house works, really. And what is giving us our driving force is not actually height, but it is active transport of protons across the cell membrane, giving us a free energy gradient which will pull the sucrose back across, which allows us to develop this pressure. This is what's giving us our pump to pull the water through. And this is one that also interests me quite a lot. This is the circulatory sugar in insects. It's another one where we have a glycosidic linkage between two anomeric carbons. In this case, we've got two alpha-glucoses. So again, this one is drawn back to front. We also find it quite a lot in fungi. But it turns out to have some really interesting additional properties in terms of the way it interacts with water. And that means that it can protect membranes and proteins and other macromolecules when water concentrations are reduced during dehydration or during freezing, because obviously one of the reasons that freezing has the effect it does is it makes water less available. So an example of this use is are in species like this. So this is actually a time series. So these plants have been allowed to dry until they go crispy, and then they've been resupplied with water, and they come back like this. And so they're able to do this because amongst the tricks they use is they produce lots of trahalo's, which means that the cells are destroyed by this dehydration process. I think you need to get one of these from my sister-in-law, who tells me that she kills every pot plant she gets. Well, she'll be hard pushed with one of these. I'm going to move fairly quickly through oligosaccharides. Because we've got quite a lot of other things to touch on, and this is not to say that they aren't important. There's an awful lot of them to enact function as elicitors and signaling molecules. And a lot of the interactions between fungi and plants, as the fungi are attacking plants, are driven by breakdown products of plant and fungal cell walls, because obviously the fungus is producing lots of digestive enzymes which are releasing bits from the plant cell walls, and the plant cells are responding to those as signals of attack. It turns out to be a lot more complex than this, and so you can use a lot of oligosaccharides and polysaccharide breakdown products as means of, for example, stimulating penicillin or other types of useful metabolic products. And in fact, a lot of research is taking place on this type of thing in the school, people like Taj Keshavats. Okay, so now we're going to come to polysaccharides. This is going to be most of the rest of the lecture. Now, starch is our main storage form of carbohydrates in plants, and we have glycogen performing a similar function in mammals. They are both primarily poly-alpha-1,4 glucose with some side branches of alpha-1,6 linkage. So what we have is a string of alpha-1,4 linked glucoses with a branch forming another chain going off at the 6 carbon of one of the glucoses, giving us a branched structure. And we have some notations here. The straight bits, when we don't have branches, are called amylose, and the more branched bits are called amylopectin. Don't confuse this with pectin, or indeed agaropectin will come too late. They're different things. And in these sections which are more heavily branched, we're seeing branches coming off every 12 to 13 residues. We're using a slightly simpler notation here, because you see I've replaced the arrows with a dash in the 1,6 linkage. That's acceptable for many purposes. You use the arrow as a sort of fully most formal version, but this is fine as well. Right, and something quite important happens here. Remember last week I spoke about amino acids differing because of their side chains, but sugars differing because of their spatial organization. And here, remember also that although we are drawing these out as flat polar projections, these are actually three-dimensional structures, and most of them will be chairs. We've got some chairs here. Okay, and what happens with the structure of starch is that because our glycosidic linkage is on the same side of both residues, and because our structure is essentially chair-like, then that structure introduces a kink. And that kink just keeps on adding. So we get a spiral structure. So this gives us a fairly open structure which allows water in, it allows enzymes in. So it means that we can easily add to and break down this structure easily. And so we have starch storage granules. As I spoke about last week, we also have specialized plastids which store starch and do nothing else. Your myeloplasts are producing starch granules in chloroplasts. And we have glycogen granules in mammalian cells, specialized mammalian cells. And again, what we've got is this spiral structure with occasionally branches coming off. It makes it a very useful structure for storing energy for short-term storage. It's got lots of water in there, it's an open structure, enzymes can get in, add to or remove. So it's a fairly quick access source of energy, which is what it stands for, which is what it's used for. I'll move on to cellulose. Again, I used this slide last week talking about how plant cell walls have as their foundation cellulose. Now, think about this. Starch is a polymer of alpha glucose and has essentially no mechanical strength. Cellulose is something strong enough to make cell walls out of. We've spoken about how high the pressures in plant cells are, how strong that material has to be. How does it differ? So this is a picture of a plant cell wall by SEM. Again, I used this last week. So what we have are neutral polysaccharides and pectins, we'll come to in a bit. But the foundation are these fibrils of cellulose. And it's useful to compare xyloglucan, which is one of the neutral polysaccharides, that so-called N-celluloses, which is beta-4-length backbone decorated with xylose intervals. And essentially, it's not possible to turn this into a solid material. It has essentially no mechanical strength at all. As soon as you add any water to it, it goes into solution, and you can't really measure it other than by viscosity. Whereas with cellulose, again, we've got the same 1,4-glucose backbone, but it is approaching as strong as steel. And the reason is that it is simple. That instead of having these glycosidic linkages on the same side of the ring, they are on opposite sides. Now what that means is not you get a zigzag linkage like this. What that means is that you get a chair conformation one way up, and then the next one goes the other way up. So the first one puts the kink in, the second one takes the kink out. So instead of forming a spiral, you get a flat ribbon. And that means that these ribbons can be aligned and hydrogen bonded together to form fibrils, hold in an extended configuration. So it's used in almost all plants and many other organisms as well. As a result, it is the most common macromolecule on Earth. And as I say, because we can get, it's a lie flat, we can get adjacent molecules, polymers, to hydrogen bond to one another to make these bundles. There's very little freedom of motion there, as a consequence of which, if you were able to measure the mechanical, if you look at the mechanical behavior of a single microfiber, and again, I'm sure many of us are wearing cellulose at the moment. If you're wearing cotton, then you're wearing cellulose, or indeed linen or what have you. And another example of how strong this can be, because it's another polymer where you get hydrogen bonds holding the material, the individual molecules together, so we get the strength of many molecules instead of the strength of one is Kevlar, which last year when I asked about people, they didn't know what Kevlar is, but it's actually what they make bulletproof armor out of, or stab vests and the like. It's an indication of how powerful these hydrogen bond structures are. It's not the hydrogen bonds that give them the strength, it's that you get ordered arrays of material. So you aren't looking at the behavior of a single molecule, it's lots of molecules forming an orderly array. And this is something from my lab. Some years I've actually brought examples of this in, but obviously I'm coming in directly, not from the lab. One of the things, the main thing I work on is plant cell walls. Okay? And what we're trying to do is work on what the different bits of the plant cell wall do. And to that end, we are making artificial plant cell walls based on this stuff, which is a bacterial cellulose produced by a species called Gluconocetrobacter. It used to be called just Acetobacter, I don't know why they changed it. So you get these, it forms these gels at the surface of the fermentation unit. And it looks really weak, it looks like agar or something. But and when I initially got the first batch of it, I was thinking it was really fragile, I was going to have trouble handling it. In fact, it was giving me difficulty to cut it with a razor blade. It is very robust material. And in fact, we've been doing some work with it as a potential biodegradable polymer as well. It is actually an SEM that an MSc student working with me over the summer produced, showing the structure of her cellulose. And this is an AFM, not from my group, showing the bugs in there as well. So you can see the Acetobacter, Gluconocetrobacter in there as well. I'll tell you a good example here. The Gluconocetrobacter should be in italic, so they tend to correct that before starting the lecture. And what it does, it's not using it as a cell wall material, it uses it as a raft. It's an obligate iron, and so it makes this layer of cellulose as a means of holding it at the surface of the medium in which it grows. Another example of these hydrogen bonded structures is chitin, which you'll see is a very similar structure to cellulose. Again, we've got a beta-1,4 linked glucose-based molecule. In this case, it's glucosamine with an alien substitution here, and we touched upon this earlier. This is used as a cell wall in fungus. It's also what insects and other arthropods make their exoskeleton out of. So this is what makes insects crunchy. Some exoskeleton instead of an internal skeleton, and that's made out of chitin. And again, what we've got are linear molecules that can be hydrogen bonded to one another to form these crystalline or partially crystalline structures which are extremely strong. the slide I used last week. This time I'm going to come back to the pectin. So here we have the pectins permeating through the wall. You can see particular rheumatology in our middle lamella here. As I said, we've got negative charges on the galacturonic acids that form a lot of the pectin, and these interact with metal ions. Since we are using osmium as a means of staining this, then we get strong interactions and strong staining of the pectin here, in the middle lamella particularly. So coming back to the slide from earlier, we have our oxidation of the hydroxyl on C6 to give us a carboxylic acid group, which, because it is negatively charged, will interact with metal ions, and with divalent ions, particularly calcium, it will cross-link the cell wall. So it will cross-link the pectins. So this has a substantial effect on their mechanical behavior. So, as I said in the first lecture, pectin is what makes the jam set. It's forming these cross-links between the pectins. This is what we're doing when we cook it up. We're forming these cross-links to make the material set. This is something from my own research, and it's something which the rest of the field are just starting to catch up with. Because for quite a long time it was believed that what determines the mechanical behavior of plant cell walls are those neutral polysaccharides and hydrogen bonding to the surface of the cellulose. And that as you pull those off, it allows the wall to extend. And it was thought that the pectins was to quite a degree irrelevant. Well, I was doing a research project, and we had said we were going to look at the levels of calcium in the cell walls. And the reason we put this in is that Matt, as then were fond of work on calcium because our collaborators have shown that calcium has quite a large effect on tomato food quality. We're looking at tomato. And one of the people I was working with wanted to play with calcium electrodes to measure the calcium levels in the water. Now he was taken off the project for one reason or another, and we didn't have anyone to do the calcium measurements. So I did this experiment to show that the calcium was unimportant. So I put in Bacta, which is a substance which binds to the calcium. This isn't a drop-in session. Well, you've been talking as well. If you want to come and go and chat to your friend, go somewhere else. You've been disturbing other people in the lecture theatre. They have paid to be here. You have paid to be here. Show some courtesy to me and the other people here. Okay? So I put in the calcium chelator to take the calcium out. And I found, so what I'm doing is stretching pieces of dark material to see what effect this has, and I was expecting to see no effect at all. And you can see that the effect of the calcium, once the stress is taking the calcium out, once the stress is applied, is actually profound. So in fact, the calcium in the cell wall, its interactions with the pectin, are actually very important. You can see that actually if we have calcium there, or if we add calcium back to the point where it saturates the calcium chelator, just to eliminate other effects that it might be having, we have virtually no effect and we have a much lower extensibility. Other people are just starting to pick up this is perhaps a potentially important effect. It's taken a good while though. I published this in 2005. Okay, hope this will work. Another thing that is becoming apparent is that plant polysaccharides in the cell walls have a substantial effect on human health. We all know very well that dietary fibre is good for the gut and that it impacts on things like colon cancer. it's becoming apparent that there are other effects as well, that there are interactions between polysaccharides, oligosaccharides coming from the plant material we eat, and our immune systems. And in fact, I've got a project student at the moment who is looking at purifying polysaccharides and is looking at their effect on different cell lines with other people within the department. I'm not going to go into this in any detail. This is another polysaccharide. Probably some of you have been showing this slide this week, or are going to. I'm not interested in the bugs. This is one of Bob's slides. I'm interested in what's underneath. The agar that we've been using to make these plates is another polysaccharide. Comes from the cell walls of red algae. They have cellulose underneath that, but kind of on the outside of that, equivalent to the hemicellulose pectins in the higher plants, we have agarose and agaropectin, which are based upon this type of monomer. Again, we've got a modified sugar in here. It's not a peculiar modification, which is a 3,5 anhydrous. So we lost a water and formed a bridge across the top of this L-galactose. Again, we've got an unusual sugar here, an L sugar, not a B sugar. And we've got a 1,4 linkage on this end, so we've got a 1,3 linkage on this end. So we go from the 1 on this sugar to the 3 on this one. And as you see, it holds water very well. It melts as you get up to about 84 degrees centigrade. It solidifies as you get down to about 40. So it provides a very useful material for this sort of for pouring gels, either for bacterial or whatever, or fungal work, or indeed for doing pure separation of DNA or RNA. Another example of a polysaccharide you come across probably a lot more than you think you do, unless you read food labels a lot. This is xanthan gum. It is a branched polysaccharide that is produced by and secreted by some bacteria. And it's used an awful lot in the food industry because of its mechanical age. What it does is it exhibits shear thinning. So you can see when your shear rate is low, then it has a high viscosity. But as you start to move it, as the shear rate increases, then the viscosity radically falls. So this is kind of the opposite. Some of you will have seen Mark Adele's oobleck, the cornflour water mixture, where you move it in sets. This is the opposite. So the place you will have come across this is if you spilt tomato ketchup on yourself. Okay? It's used to thicken a lot of sauces and dressings and things, and other processed foods. So what it means is that when you start to pour the tomato ketchup, it is solid. It doesn't do anything. You start to shake it, and then all of a sudden it starts to shear and flows. And so it will then go everywhere. Why do you think this is a good idea? I don't know. But what is actually happening in this case is it is highly branched, and when it's still, then those branches are entangled, but as it starts to move, then the branches get pulled and sheared back against the backbone of the molecule, and it becomes streamlined and shoots through. Okay, last thing I'm going to say before I finish, I'm going to mention the importance of sugars to proteins. Now, in bacterial proteins, this doesn't happen. So bacterial proteins are just polypeptides. But we have a number of modifications that can happen after eukaryotic proteins are produced. And one of the most common, and it seems to happen to an awful lot of them, are addition of sugar groups. And they can either be added to an asparagine, in which case they're N-linked because they're being attached to a nitrogen, or a serine or a threonine, in which case they are O-linked. This can be quite a substantial problem in working with proteins, recombinant proteins, because a lot of them, a lot of eukaryotic proteins, when you express them in E. coli or whatever, don't work. This is simply because they don't have the sugars put on them, and so that they are not fully functional. And in many cases, you're talking about proteins which are more sugar than they are protein, in fact. And the other really important issue in terms of these molecules, of this, is that in eukaryotes, then glycosylation is often used to target the proteins to the correct subcellular location. So they're used as providing signals to tell the cell where to take, to direct where the proteins go. So you add a particular group of sugars, and that says it's got to go to this organ now. And the other very important set of issues, a lot of our cells have, all of our cells are decorated with glycoproteins. So we actually have a surface layer of sugars to our cells. Now, Miriam Dweck in this school is doing some very important work on this. What she is looking at are distinctive glycosylation patterns on the surface of cancer cells. So this allows you to potentially identify the target cancer cells, and particularly to tell between different types of cancer. Because even within an organ, you have some types of cancer which are relatively treatable and others which are serious problems. And being able to identify which you've got at an early stage is potentially very useful. She's working on ways that this can be done, particularly with breast cancer. But the place this was found earliest is in our blood groups, because each of us has a different pattern of glycosylation on the surface of our red blood cells, and that means that our antibodies are primed to attack any other pattern of glycosylation. It was a big puzzle when people started to try and carry out blood transfusions, why sometimes it worked fine, and sometimes the person would keel over due to it. And of course what it turns out is that what this is an immune response because of the wrong type of glycosylation in blood cells. Okay, I will finish there. Give you a break until about 10 when Caroline Smith will be picking up. I'm not sure what she's teaching, but it'll probably be something very related. Thank you very much.